Here we're going to look at enzymes. Enzymes are a globular protein, so that means they are functional. And their function is to be a catalyst. This means that they speed up the chemical reactions in your body. Rx stands for reaction. So you have constant chemical reactions going on in your body. Remember, chemical reactions are building or breaking bonds to build or break molecules. So you either build or break molecules by forming or breaking bonds. And enzymes speed this up. So you rely on these chemical reactions to keep you alive. And they would happen on their own without enzymes, but they would happen so slowly that they wouldn't keep you alive. You would die waiting for those reactions to occur. So you need enzymes in order to make the reactions go fast enough to keep you alive. So remember, reactions occur in two types. You can have decomposition, also, if decomposition occurs in your body, it is called catabolism. So if we look at what's going on here, here we have lactose. You've probably heard of that. Lactose is the sugar in milk. It is a disaccharide. So di means two. Notice it's two individual carbohydrates linked together. It's glucose and galactose linked together to make a disaccharide. Here you have the enzyme lactase. Lactase binds to the lactose. So in this case, lactose is our substrate. The substrate is the molecule that the enzyme acts on. Notice that the enzyme has an active site, and it is shaped to fit the substrate. So enzymes are specific to which substrate they'll bind to. So think about people who are lactose intolerant. That means they can't digest the sugar in milk. People who are lactose intolerant don't have the lactase enzyme. So when they drink milk, they can't digest the sugar in it, and that sugar goes through their intestines undigested, and that's what causes the diarrhea and gas and bloating and cramps. So you have to have an enzyme for each particular type of food that you eat in order to digest it. So notice that the substrate binds to the enzyme. So here they bind to each other. The substrate fits right into that active site. Then next, the enzyme bends. If you look closely, look at this picture, look at this picture. See how the enzyme has a different shape? That's the key to how enzymes work. Enzymes work by bending. When it bends, notice that that bond breaks. Now, instead of a disaccharide, we have two monosaccharides. The molecule or molecules that you form are called the product. So our product now is one glucose, one galactose. And then notice the arrow goes back around here. You're ready to reuse that enzyme. It's ready to go again. Down here, we have the opposite reaction. These are individual glucose molecules. Glucose is blood sugar. So whenever you eat carbohydrates, you end up with glucose in your blood. If you eat starch or you eat sugar, whatever it is, your digestive system converts it to glucose. If you have extra glucose in your blood, you store it as glycogen in your liver and muscles. So this is an enzyme found in your liver and in your muscles that takes extra blood glucose and links them together to make a glycogen. So here you can see the glycogen is just several glucose molecules linked together. So this is a synthesis reaction. And when synthesis reactions occur in the body, they are called anabolism. So it's the same thing. Glucose is going to be the substrate. Notice how the glucose molecules fit into the enzyme active site. When they bind, this is called an enzyme substrate complex. The same up top here, when they bind, that is also called an enzyme substrate complex. Then the enzyme bends, so if you look at the enzyme here compared to here, you can see that the enzyme bends. And this time when it bends, we form a bond. And you get glucose linked together so that you get glycogen. Some important factors affect enzyme rate. So when we think about enzymes, enzyme rate is very important. Remember, these chemical reactions have to occur at just the right speed in order to keep you alive. So enzyme rate, one of the big factors that influence it is temperature. If it is too cold, the enzymes are stiff. Think about like Play-Doh or plastic, something that if it gets cold, it's stiff. And remember, enzymes work by bending. So if they're cold and stiff, they don't bend well. And this slows them down. Think about how you put your food in the refrigerator. If you had leftovers and you left them sitting out, they would be rotten by the next day. If you put your leftovers in the refrigerator, you can keep them for a week because there's bacteria that rot your food. And the bacteria, like any living thing, use enzymes to rot your food. If you put your food in the refrigerator, the cold slows those enzymes and the bacteria is not able to rot your food very quickly and your food stays good for a long time. If you warm enzymes up a little bit, they will be more flexible and they will work faster. This is what happens when you have a fever. So there's kind of this knee-jerk reaction that as soon as people get a fever, they think, oh my, I need Tylenol or Advil or something. That's not really true. A fever is good for you. It's part of your body's defense. Your body uses the fever to fight the infection. And if you're taking Tylenol and Advil to get rid of the fever, all you're doing is fighting against your own body's defense against the infection. You're working against yourself when you do that. So fevers warm your enzymes and make them work faster so that you can fight the infection faster. Now, there is a catch though. You don't want to get too hot. So a fever that starts getting up to 104, then you do want to get the Tylenol and the Advil and start treating it. Because if you get too hot, you run the risk of denaturing your enzymes. So proteins are denatured if they get too hot or if you change the pH. And if you denature them, they don't work at all. And you need your enzymes to stay alive. So always listen to your doctor when it comes to a fever, but in general, a low fever is helping you as long as it doesn't last too long. If you've had it for a long time, go to the doctor. If your doctor says you need to treat the fever, then of course do it. So in addition to temperature, pH is the other important factor for helping your enzymes work. And remember, we talked about before that your blood pH has to stay at 7.4. So it's because of your enzymes is one of the reasons why this is so important. If this gets too high or too low, if it changes in either direction, you will denature your enzymes. And again, you can't live if you denature your enzymes. So maintaining temperature, maintaining pH are a couple of important things in order to keep your enzymes functioning well so that they can keep those chemical reactions going and keep you alive.